Marsilea, a pteridophytic genus commonly known as water fern, usually grows in mud, shallow ponds and pools with the leaves floating on the surface of water. Its species are ubiquitous, that means they are distributed throughout the world but are more common in tropical Africa and Australia. The genus is represented world over by 53 living and 10 fossil species. In India, Marsilea is represented by 9 species. Of these 9 species, Marsilea minuta and Marsilea quadrifolia are most common and widely distributed one. Its systematic position. The Marsilea belong to kingdom Plantae, division Pteridophyta, subdivision Pteropsida, class Phelicophyta, order Marsiales, family Marsiaceae, and genus is Marsilea. The plant body of Marsilea is sporophytic, having diploid chromosome number in its somatic cells. The sporophyte is differentiated into rhizome, roots and leaves. We will first discuss about the rhizome. The stem is long and slender. It either creeps on the surface like stolons or slightly below the surface of the soil, just like rhizome. It is grow extensively and is branched. The stem is divisible into distinct nodes and internodes. The internodes are long in case of hydrophytic species, that means those species which grow in water bodies, and they are short in subterrestrial or xerophytic species. Then we'll discuss about the root. The primary root formed on the stem, it is short-lived and it is replaced by adventitious roots which arise gradually at the nodes on the underside of the stem. Then it is leaves. The leaves arise from the upper side of stem and are arranged in 2 alternate rows. They are long, petiolated and compound. The leaf lamina is usually divided into 4 leaflets of the same size. They spring from tip of the petiole so that the leaf apparently looks like quadrifoliate. At the base of the petiole, many bean-shaped or oval and stalked structures arise, which are called as spore corps. When we cut the transverse section of the Marsilea, it shows certain distinguishing features. First we will discuss about rhizome. When we cut the transverse section of the rhizome, it shows certain features, like the outer covering is called as epidermis. It is single-layered, made up of compactly arranged thick-walled cells. Then epidermis is followed by cortex, which is differentiated into 3 regions in case of Marsilea, that is outer cortex, middle cortex and inner cortex. The outer cortex is aerenchymatous. It is made up of several cells in thickness having large intercellular spaces. These aerenchymatous cortex, they help the plant to float on the surface of water. Then it is followed by middle cortex, which consists of sclerenchymatous tissue, and then inner cortex is solid tissue made up of several cells in thickness. Then the vascular bundle which is present in the center, it is siphonostele. It is limited externally and internally by endodermis, hence called outer endodermis and inner endodermis respectively. And the siphonostele in case of Marsilea is medullated, which means it has a pith at the center of the stem. The xylem is in the form of ring and phloem is present on both sides of the xylem. Such a stele in which phloem is present on both sides of xylem is called Amphiphloic-siphonostele. Then we discuss transverse section of the petiole, means the stalk of the leaf. It shows single-layered epidermis made up of rectangular cells. The outer cortex consists of 5 layers of thin-walled cells. Middle cortex is erenchymes, consists of large number of air chambers, while the inner cortex is solid, compact tissue of several cells deep. In case of pteridophytes, the vascular bundle is called as setile. It is covered externally by single-layered distinct endodermis. It is triangular in outline, lies in the center, and has a single vascular bundle. The xylem that is present in the center is V-shaped with openings towards the axis. Then, when we cut a transverse section of the leaflet, it consists of upper and lower epidermal layers, each made up of single layer of parenchymatous cells. The continuity of epidermis is interrupted by sunken stomata. Stomata are usually restricted to the upper epidermis in case of floating leaves. Then the ground tissue, that is mesophyll, it is present in between the upper and lower epidermis and is differentiated into palisade and spongy parenchyma. The palisade part, which lies just beneath the upper epidermis, it consists of columnar cells rich in chloroplasts, while as the spongy part, which faces the lower epidermis, it consists of rounded cells. However, in case of submerged species, there is no distinction between palisade and spongy tissue. Vascular bundle in case of this leaf they are embedded in the mesophyll tissue. They are concentric in nature, which means that xylem is surrounded by phloem. Then when we cut a transverse section of the root, it shows the pelliferous layer instead of epidermis, made up of compactly arranged biconvex cells. Beneath this pelliferous layer, the cortex is made up of outer cortex and then inner cortex. The outer cortex is erenchymes. It consists of large air cavities or air chambers which are arranged in the form of ring. The inner cortex is compact, made up of rounded cells containing starch. And the inner cortex is delimited by a single layer of distinct endodermis. A vascular bundle lies within the endodermis and is usually diarch and exarch. The xylem is plate-like and occupies the center of the setile. That means protoxylem consists of small mass of cells and is lying towards the periphery, that means towards the pericycle, while as metaxylem, which consists of large number of cells, it is made up of plate-like and occupies the center of the leaf. Marsilia reproduce by vegetative as well as sexual means. Vegetative reproduction in case of Marsilia, it occurs by the formation of tubers, which are small bud-like structures containing reserve food material arising from the subterranean branches of the rhizome. Then I will discuss about the sexual reproduction. In Marsilia, since it is a heterospore fern which produces two types of spores, that's microspores and megaspores, and they are produced in a separate sporangia born in special bean-shaped bodies called the spore corps. Usually, one spore corp is present at the base of each petiole, but in some species, the number varies from 2 to 20. Sometimes the attachment of spore corp with a petiole shows so much variation that different species can be distinguished or delimited on this particular character. For example, in case of Marsilia polycarpa, many spore corps are attached on one side of the petiole in a single vertical row. In case of Marsilia quadrifolia, stalks are united with one another and then jointly inserted on the petiole. Then in case of Marsilia minuta, the stalk of all the spore corps are though free but are attached to the petiole at a single common point. Then we will discuss about the structure of spore corp. The spore corp is differentiated into a stalk and a body. The stalk is fused laterally to the back of the body of spore corp, generally forming a distinct structure or ridge called as a raphe. Then sori. When we cut the horizontal section of the spore carp, a ring appears in the form of dorsal and ventral mass. In this plan, both micro as well as megasporangia are visible as sori. These sori are the reproductive structures arranged in two alternating rows in the cavity of the spore carp. It is surrounded by an inducium, that is a layer which surrounds this sorus. The number of sori in a spore carp varies from 2 in case of Marsilea aegyptica to 20 in case of Marsilea quadrifolia and Marsilea vestata. The number of micro and megasporangia varies within species. In Marsilea minuta, a source has 4 to 8 megasporangia and 8 to 13 microsporangia. Then development of sporangia, how it develops. Since Marsilea is a heterosperse fern, microsporangia produce microspores and megasporangia produce megaspores. The sporangia develop in a basipetal manner on the receptacle, that is the long axis of the sporangia. The megasporangia develop first at the top of receptacle and are therefore older one, while the microsporangia develop later along the sides of the receptacle. Then development of microsporangium as well as megasporangium is almost similar in all the species of Marsilea. How it develops, how the development of the sporangia takes place. One of the cell formed at the apex of the receptacle functions as megasporangial initial. It divides transversely, forming an outer cell and an inner cell. The entire sporangium develops from the outer cell and inner cell, taking no part in its development. In megasporangia, all spores degenerate, but only one remains. These degenerated spores, they form a nutritive tissue, and the spore which remains, they grow in size at the expense of the others, forming a single large functional megaspore. Subsequently, two initials are formed on the lateral sides of the receptacle. They function as microsporangial initial, each forming a microsporangium. Then the outer cells of the sporangium, that means the cell which is destined to form spore mother cell, undergo a repeated transverse division, and each spore mother cell, it undergoes reduction division and form 4 haploid spores. In microsporangia, almost all the spores in a microsporangium are functional, and they are smaller in size than megaspores. Then dehiscence, how they liberates, how these spore carps liberates. The dehiscence of spore carp. There is no special mechanism for the liberation of sporangia from the spore carp. However, they set free only when the wall of spore carp splits open. As the spore carp breaks at one point due to absorption of water, the sori come out along the gelatinous ring of the spore carp. And these sori, they function as mother cells of the gametophytic generation. Then gametophytic generation, how it starts. The microspores and megaspores, they germinate separately to produce male and female gametophytes. Structure of spore carp. The spore carp is differentiated into a stalk and a body. The stalk is fused laterally to the back of the body of spore carp, generally forming a distinct structure or ridge called as a raphe. Then dehiscence, how they liberates, how these spore carps liberates. The dehiscence of spore carp. There is no special mechanism for the liberation of sporangia from the spore carp. However, they set free only when the wall of spore carp splits open. As the spore carp breaks at one point due to absorption of water, the sori come out along the gelatinous ring of the spore carp. And these sori, they function as mother cells of the gametophytic generation. Then gametophytic generation, how it starts. The microspores and megaspores, they germinate separately to produce male and female gametophytes. When one of the sporum, that is androcytes, it fuses with the egg to accomplish fertilization and form the diploid zygote. The zygote acts as a mother cell of the next sporophytic generation and develop into young sporophyte within 2 to 4 days after fertilization. Then embryogeny, when the fertilized egg, that's called as zygote, it enlarges in size and secretes a thin cellulose wall around it. This zygote, it divides vertically and produces 2 unequal cells. The smaller cell is called as hypobasal cell and the large cell is called as epibasal cell. These 2 cells undergo again transverse division to form quadrant embryo. That means embryo consists of 4 cells. The cells of the quadrant embryo shows definite relation to the primary growth of the embryo. Then epibasal half produce shoot and leaf, while as hypobasal half it produces root and the fruit. Such a development where epibasal half produce shoot and leaf and hypobasal one produce root and fruit is described as lateral. Then at the time of differentiation of different primordia in embryo, the adjoining cells of megagametophyte divide periclinally, forming a thick calyptra which encloses and protects the developing embryo. The calyptra is greenish in color. A large number of rhizoids may develop from the base of the calyptra. The embryo gives rise to a young seedling that penetrates through the calyptra and grows into a complete sporophytic plant. Life cycle of the Marsilea, we find alternation of generations, which means that Marsilea plant is a sporophyte which is dominant phase in its life cycle, contrary to the in case of bryophytes, where the dominant phase is haploid in nature, whereas in case of pteridophytic plant in which the Marsilea is placed, the dominant phase is sporophytic. This sporophyte comprises rhizome, root and leaves. It reproduces vegetatively as well as by means of spores. The spores are formed in Sporangia on specialized structures called spore corps. These spore corps bear 2 kinds of spores which are formed after meiosis. Hence they are also called as haploid meiospores. The smaller spores are known as microspores. They are produced within microsporangia, while as the largest spores are known as megaspores, which are formed in megasporangia. The microspores and megaspores, it germinate separately to produce male and female gametophytes. They bear antheridia and orchigonia, which produce anthrozoids and egg respectively. The fertilization of male and the female gametes gives rise to embryo and ultimately develops into sporophyte. And thus once again starts its life cycle.